Emily Isco, recently Dr. Isco, I guess. I'm a graduate student at Cold Spring Harbor. I'm in my 6th year. My project has been comparing the neural circuits between lab mice and singing mice and asking questions of the evolution of neural circuitry. Most labs study the lab mouse, Mus musculus, which is the same species of mouse that if you ever had a mouse infestation in your house, it's the same species, and that's the species that most scientists study. Here we have a male and a female lab mouse in a cage together. When they're together in close contact like this, that's when they make these quiet vocalizations that we call ultrasonic vocalizations, or USVs, because they're so high-pitched they're outside the human hearing range. The graph at the bottom is what we call a spectrogram. The x-axis is time and the y-axis is pitch or frequency, and the dark squiggles are their vocalizations. In our lab, we study an additional species that we call the singing mouse, or Scitinnimus toguina. So that's a species of mouse from Costa Rica. And what's special about those mice is they have very unique vocalizations. So this one shows the same setup, but instead of lab mice, we have the singing mice. So we see two vocalization types, a much quieter type that we believe is akin to the lab mouse USVs, and a much louder vocalization type that we refer to as singing. The softer USV vocalization type is common to basically all rodents, and this louder singing has emerged more recently in the singing mice. You can ask a lot of questions about how the brain kind of supports this interesting vocal behavior. In my project, we focused on the evolutionary aspect. Singing mice sing, lab mice don't, so what in the brains kind of changed and evolved in order to evolve this novel behavior? We hypothesized that perhaps it was a difference in the structural circuitry, specifically in this cortical region, the region of the motor cortex that we knew was involved in controlling the singing behavior. So this experiment proved that all the major targets of these neurons were the same between the two species. But it's really hard to quantify and look at individual neuron projection patterns. To address those issues, we use a technology called MapSeq. And so the way that MapSeq works is we use unique RNA barcodes. So each neuron is labeled with a unique RNA barcode, and you can dissect out different target regions, extract the RNA and sequence it, and know exactly which neurons project to which region, because we can detect all the unique barcodes in a downstream region and match that to the soma of the originating neuron. Coming into the study, we weren't really sure what we were going to find. We thought maybe there would be kind of a dedicated song circuit. We would look at the brains and there would be some brain region that is just completely different between the two, but that's not what we found. Instead, we find that it's not the case that there's a novel circuitry, a novel connection. It's that it's kind of a tweak on a pre-existing circuit. There's a greater proportion of neurons that project to certain downstream regions in the singing mouse. Even these very specific changes within a circuit can cause drastic effects on behavior. If you want to change a behavior very rapidly on a much shorter timescale, tweaking an existing circuit is kind of an easier avenue for evolution than coming up with a brand new plan. This approach that we've used of using two closely related species with divergent behavior, identifying a region that is causally involved in the behavior, and then applying MapSeq to that region and finding a significant evolutionary difference between the species. We think that this approach can be applied to other model organisms, other species. Other labs can try to also identify circuit differences involved in the behaviors of the species that they're interested in.